Hello, we are from Group 1. We're presenting to you our PBL report for AMJ40503 Environmental Remediation Subject. So the title of our study is the study on copper 2 ions removal efficiency on different types of aquatic plants and operating condition. So I will introduce myself. My name is Eugene Daniel Crimson and I will talk about our background study. So first of all, phytoremediation is one of the biological-based technology that has been proven successful in treating contaminated water in recent years. Phytoremediation is the process of using plants and associated soil microorganisms to lower the concentration and toxic effect of contaminant present in the environment, such as soil, surface water, and groundwater. So basically, this study is focused on removing heavy metal from the water contamination phase by using aquatic plants. Heavy metals refers to any metallic chemical element with a relatively high density and will cause environmental and health risks at high concentration. Phytoremediation has several advantages over traditional physical and chemical cleanup approaches. First, it is low in cost. Second, it is environmentally friendly. And third, it is applicable for a wide range of contaminants. So, in order to achieve high effectiveness in phytoremediation, plants with high capacity for heavy metal accumulation and long and hairy roots are the best choice. In this research, water hyacinth and water thyme were chosen as the study target for copper 2 ions removal efficiency. In addition, the effects of contact time and initial concentration of copper 2 ion presence in a water sample were also investigated in this study.
Hi, my name is Fakhrul bin Samah. So this is problem statement in this study, which is, as we know, heavy metal pollution is a serious problem which is contributed to harmful effect on human health. Due to their high solubility in the aquatic environment, the discharge contaminated heavy metal in the wastewater can observed by living organisms. Once once the contaminated wastewater enter the food chains, the heavy metal may accumulate in the human bodies. It may contribute to serious health disorder, which is include reduced growth and development, cancer, organ damage, central nervous system, and in the worst term is death. So therefore, remediation technology is now widely used in the removing of heavy metal from the contaminated wastewater and soil. Phytoremediation refer to a broad concept of utilizing environmental technology based on green plant on removing heavy metal from water, soil, and sediment. The next objective of this study is to compare the copper ion removal efficiency of different type of aquatic plant in water sample. Second, to study the effect of different operating parameter in terms of contact time and initial concentration on the copper ion uptake in the water sample. Third, to conduct the physical monitoring on the wetland plant condition after phytoremediation of heavy metal from the water sample.
Hi everyone, my name is Chong Jing and I'll be discussing about the methodology part for our PBL. As you can see from the slides, this flowchart indicates the overall experimental flow for our work. These are the materials used, where copper 2 sulfate pentahydrate are used to supply copper ions to the water samples. Next is on the preparation of phytoremediation plants. Two types of aquatic plants were studied in this PBL, where water hyacinth was collected from Tasik Malaki, while water thyme was bought from aquarium shop. Sufficient amount of origin water where these aquatic plants grow was collected for acclimatization purpose. Next is on the sample preparation and setup. Firstly, plants were washed thoroughly with tap water followed by rinsing with distilled water to remove contaminants. Next, 200 ml of origin water was mixed with tap water in a container. Ammonium nitrate was added into the water sample to provide nutrient for plant growth. Then, the plants were placed under sufficient sunlight for 5 days before start of experiment. There are two main objectives for our PBL, which are study on effect of contact time and initial copper concentration. For the study on contact time, 100 grams of water hyacinth and water thyme were placed in separate container containing 10 mg per liter of copper. Meanwhile, for the study on initial copper concentration, 100 grams of water hyacinth were planted in 3 sets of water sample containing 5, 10, and 50 mg per liter of copper. Both experiments were lasted for 15 days, where water samples were collected in a 3 days interval for data analysis using atomic absorption spectrometry. There are several calculations involved in the methodology, and the first one is molar equation, where it was used to prepare copper stock solution. Next, dilution equation was used to dilute the copper stock solution into required concentration of copper containing water sample. Lastly, heavy metal percentage removal was calculated based on this equation after the data were collected. That's all from me. I will now pass it to my teammates to discuss on the results and discussion part. Thank you.
Good day to Doctor. I'm Yi Ting from Group 1. Now I would like to talk about the results and discussion for Objective 1. The comparison of different aquatic plants, which is floating vs submerged plants. The Hydrilla and Econia crab sites were used to treat 10 mg per liter of copper 2 ions for 15 days. The table shows the removal efficiency of copper 2 ions by Hydrilla and Econia crab sites. Figure A shows the concentration, while Figure B shows the removal efficiency of 10 mg per liter of copper 2 ions by Hydrilla and Econia crab sites within 15 days. The copper 2 ion removal efficiency for both Hydrilla and Econia crab sites has increased drastically from day 0 to day 3 and has reached their peak in day 4, where the removal efficiency has attained within the range of 70 to 80% thereafter until the end of the experiment, which is day 15. This may be due to the Hydrilla and Econia crab have a high level of capacity to accumulate the copper 2 ions initially due to the presence of available sites. When the capacity has cracked and has reached an optimum level, when the process of uptake for copper 2 ions will be slowed down. For day 4 to day 15, the copper 2 ions removal efficiency for Hydrilla and Econia crab has dropped from 72% to 70.97% and 79.43 to 76.84% respectively. The decrease of removal efficiency after day 4 may be due to evapotranspiration process. This is because the water sample might have evaporated at a higher rate due to hot weather. As the volume of sample has decreased, the concentration of copper 2 ions will increase. When the concentration of copper 2 ion increase, it will cause the decrease in removal efficiency of copper 2 ions by both of the aquatic plants. In order to get a more accurate results, small amount of distilled water shall be refilled to the sample solution every time after water sample are collected. For Hydrilla, the copper 2 ion removal efficiency after day 4 was maintained within the range of 70 to 75%, while for Econia crab it was remained within the range of 75 to 80%. Based on the result obtained, it can be clearly seen that the overall removal efficiency of copper 2 ions by Econia crab which is 76.84%, is higher than Hydrilla, which is 70.97%. This may be due to the Econia crab has longer and hairy root system that is able to increase the potential to scour copper 2 ions. Besides, the Econia crab also has very thin root cuticles, which is capable to extract copper 2 ions through the entire surface of water. That's all for my part. Thank you.
Greetings, I'm Frankie from AMJ40503 Environmental Engineering. I'm going to present the result and the discussions on the effect of the contact time and concentration of Echronia crab size for copper-2 ion extractions and the conclusion of our PBL projects. The figure shows the copper-2 ions in water sample of Echronia crab size with different initial copper-2 ion within 15 days. At the early stage, the concentration of copper-2 ions for all the 3 water samples decreased drastically from day 0 to day 3. From day 6 onwards, the concentration of copper-2 ions for all 3 water samples have fluctuated within a small range. For all the 3 samples, the copper-2 ion concentration have shown a same trend, where the copper-2 ion concentration in water sample has increased slightly on day 15 as compared to day 3. However, it can be seen that the general copper-2 ion concentrations for all 3 water samples have shown a downward trend when the contact time increased. This trend may be due to the high capacity of Echronia crab size to uptake copper-2 ion at the early stage. Once the heavy metal uptake capacity has reached an optimum, the final concentration of copper-2 ion will not fluctuate much. Hence, it can be concluded that the removal efficiency is directly proportional to the contact time. The figure shows the percentage removal of Echronia crab size in different concentrations, which are 5 mg per liter, 10 mg per liter, and 15 mg per liter. For the study on effect of concentrations, 3 different water samples containing different copper-2 ion concentrations were prepared based on the procedure stated in methodology parts. Based on the graph plotted, copper-2 ion with concentration of 15 mg per liter has the highest percentage removal as compared with the others. The result can be explained by the heavy metal bioavailability in the experiments. When the initial concentration of the heavy metal increase, the bioavailability also increase. Thus, more heavy metal are available in the water sample to be absorbed by Echronia crab size. Hence, it can be concluded that heavy metal removal efficiency is directly proportional to the initial heavy metal concentration. So, in conclusions, phytoremediation is an effective method to remove heavy metal from water by using aquatic plants such as Hydrilla verticillans and Echronia crab size. Our study have shown that the overall removal efficiency of 10 mg per liter of copper-2 ions by Hydrilla verticillans and Echronia crab size is about 70.97% and 76.84% respectively. Based on the result obtained, it can be clearly seen that the overall removal efficiency of copper-2 ions by Echronia crab size is higher than Hydrilla verticillans. On the study of effect of initial heavy metal concentrations, the removal efficiency of Echronia crab size with 3 different concentrations was recorded, which are 51.80%, 76.84%, and 82.73% respectively. From these experiments, we have identified that the removal rate of copper-2 ion at concentration of 15 mg per liter was the highest. It is because the initial concentration of heavy metal increased, the bioavailability also increased. Therefore, we can conclude that heavy metal removal efficiency is directly proportional to the initial heavy metal concentrations. Hence, it is recommended that phytoremediation technologies can be transferred from laboratory to practical applications on a large scale in the future. That's all for our presentations. Thank you.